Greetings, ladies and gentlemen. Mr. Sanger live from the storeroom here, and we are going to talk about a lab called the Fruit Fly Behavior Lab that goes with our ecology unit. So, um, you are actually going to design your own experiment in this case to answer your own scientific question. We're going to review how to write a lab report. It's been a while since we've done a formal lab write-up. So we're going to review that and then what elements should be included in each section. You're going to apply statistics, specifically standard deviation and standard error, to analyze your results. And then finally, the content piece of this is to explore how environmental factors affect the behavior of an organism. To be honest, this really is more about process and doing science and less about, the specific question that you ask or answer. We're not as concerned about that. So to design your experiment, so to study behavior, we are actually going to be using wingless fruit flies. These are available at some pet stores. They're basically fruit flies that can't fly because they have vestigial wings. Maybe you remember the vestigial wing trait back when we looked at, did some genetics problems with them. So these are fruit flies with these tiny itty bitty wings that don't actually work, so they can't move around, which means lizards have an easy time finding them and eating them, which is sad. But they're also kind of useful for this particular behavior lab, which works out well for us. Now, to collect them, what's going to happen is you're going to get a little, condiment container here. Can't get that centered. And I'll just kind of put, 20 or so fruit flies in here for you. And then you're going to need to get them into a straw. And so to do that, you're going to create a little device here. I want to call it the suckometer, but I don't know, is that inappropriate? I don't know. But basically, there's a straw here with a little bit of gauze. And what you can do is you pull your flies and you go, and you like suck them up into your straw to get them in there. And then once they're in, you can sort of close off the end with a Q-tip. And then you can pull the gauze off and replace the other end with a Q-tip. And that is going to be your choice chamber as indicated in this picture. So you're going to basically place 20 fruit flies into the choice chamber. And then the data you're going to collect is how many fruit flies are on each end every 30 seconds for 5 minutes. So every 30 seconds, you're going to do a count. If the center line is here, you're going to be like, okay, there's 13 on this side and there's 7 on that side. And then 30 seconds later, okay, now it's 15 and 5. And you're going to sort of track that data over time. Once you do that, you're then going to manipulate a variable of your choice to see how it affects where they go. So the first trial is going to be your control trial, where you're just going to use distilled water for each of your 2 Q-tips. So again, you're going to have a Q-tip on one end, you're going to have a Q-tip in the other, and in this first trial, both Q-tips are just saturated with distilled water. So theoretically, there should be no preference either way for these fruit flies. It's really more, how do they just move around when there's no choice? So that's the purpose of the control. For the experimental trial, this is where you get to make some decisions. You could choose to investigate what is called chemotaxis, which is a response to a particular chemical. So you could use chemicals, dip this in, say, a sugar water solution or a salt water solution, vinegar, fruit, condiments, cleaner, something like that. Nothing that will harm the fruit flies, please. Phototaxis, you could look at different lighting conditions. So maybe you create a dark side and a light side and see if that has any impact, or a red side and a blue side using transparent like cellophane, which is this colored saran wrap essentially. Geotaxis, maybe you mount this thing at an angle to something or even vertically and see, do they care where they go? So you can be creative with this. Again, be sure you take good care of our fruit flies. But so long as you're not being cruel or unusual in your punishment of them, then you can have pretty much free reign. The only real thing is make sure you consider which treatments could give you interesting insight into the behavior of your fruit flies. So we don't want it to be just totally random. You want to try and answer a specific question. And even better, like, why would that have evolved? So Okay, so in terms of writing your lab report, just a reminder on Schoology, there is information on how to write a lab report and how to graph using Google Sheets. So if it's been a while since we've done this, so it may be worth referencing those documents. Here's kind of the gist. Before the lab day, you're going to do a pre-lab, which will include a purpose. This is where you state the specific question that you are trying to answer. So this is not, we're trying to learn about the behavior of fruit flies. This is, we will determine whether fruit flies prefer sugar water or salt water, or whether they prefer red light or blue light, or whatever it is. Okay, so very specific. Your hypothesis is where you predict and explain. For your methods, you want to show me what 4 tests you performed. So we're going to ask you to do 4 different tests, 1 control, where it's just distilled water, and then 3 additional tests. And think carefully about what those 3 are. So, if you're testing sugar and salt, you might want to do like sugar v water, salt v water, and sugar v salt. And that way you can see, like, if it's just sugar is a choice, are they drawn to it? Are they repelled to it? And that sort of thing. So again, you want to try and answer the question as much as you can. So I think 4 tests should help you do that, but it means you have to be very specific in what you're going to do with those. So you're just going to list them, draw a picture, table, whatever is easiest, but just kind of communicate to your teacher what your tests are. And then the results for your pre-lab, you're going to make a blank data table for how many fruit flies were on each side of the chamber every 30 seconds for 5 minutes. And you're going to include space for your control and each of your 4 tests. So we're going to let you guys in your groups try and figure out exactly how to make that work. So then we're going to do the lab, and then you're going to do a post-lab. And before I get to that, just a reminder, the post-lab has results and conclusions. So your results should have your completed data table. And then you're going to actually do a chi-square analysis for, actually, you know what? We're going to skip that. Just kidding. Skip the chi-square. We're not doing chi-square. But you are going to need graphs of the average number of fruit flies on each side of the chamber for each of the 4 tests with standard error bars. And then finally, for conclusions, you're going to want a claim evidence reasoning analysis where you do your best to try and explain why your fruit flies might have shown the preferences they did, and then sources of experimental error. And for this one, those should be bountiful because we're dealing with fruit flies who don't always behave the same way and do strange things sometimes. So that's kind of the gist. Now, at this point, I'm going to review error bars and just how those work. So if you feel like you've got that, you can calculate them, you know how to do standard deviation, standard error, you could probably stop listening right now. But if you've always been a little on those error bars and standard deviation, standard error, maybe keep going. And a lot of this, it is review of previous stuff, but it's probably worth it. So, so the question is, let's suppose, oh, there's the bell. You did side A, and you just recorded how many fruit flies were on side A during trial 1 and trial 2. And when you finished, you got the average of 11.4, and you graphed it. So side A for trial 1 had an average of 11.4 fruit flies. Side A for trial 2 had 11.4. So you might look at this and think, okay, so is that exactly the same? Is there any difference in this data? But remember, the average is just one way to describe a set of values. We also have the standard deviation and we have the standard error. And those are important pieces to include in the conversation. So, so error bars kind of help us get at that a little bit. So, so let's talk about error bars. These help us to see if differences are statistically significant or not. So if the error bars overlap, that means there is no statistically significant difference between these groups. So in this case, the control and treated are not statistically significantly different. In this case, though, the error bars do not overlap, so we would say there is a statistically significant difference between these 2 treatments. So what are these error bars and what are they showing? So the error bars are actually 2 times a value called the standard error of the mean. To calculate the standard error and to produce your error bars, you have to do a few things. You need to first calculate what's called the standard deviation. Then you need to actually calculate the standard error. And then since the error bars are 2 times the standard error, you need to multiply that standard error by 2. So your formulas and equation sheet for AP Biology has the equations for standard deviation and standard error. And so you can calculate that. So let's start with standard deviation. This is simply a measure of the spread of the data. How much variation is there? So in any group of data, there's going to be a range. Well, not any group, but if there's a range of data and the mean is here, we sort of set up this normal distribution. And we say that one standard deviation is defined as encompassing 68.3% of the data. So on either side, it's like 34.15 and 34.15% of the data is within one standard deviation. 2 standard deviations will encompass 95.4% of the data. 3 standard deviations encompass 99.7% of the data. So we got our mean and then our different standard deviations. So it doesn't matter how tight or how wide the curve is. No matter what, 3 standard deviations will encompass 99% of the data. So if you have, most of your values are all really close to the mean, you have a nice tight distribution like this. And if the values vary a little bit more, then you're going to have a wide standard deviation like that. So again, the equation for calculating this is on the formulas and equation sheet. And you basically just, plug these numbers in. So your data value minus the mean squared over the number of trials, which in this case would be 1, 2, 3, 4, 5, because that's the mean, minus 1. And if you did that correctly, you could try it out in practice. You should get these values. And so you might think, well, these 2 trials are the same because they have the same average, but you can see inside A, it's a much tighter standard deviation than in trial 2. So there actually is a difference between these 2 data sets. There's a video here where someone else explains standard deviation if you want that. Standard error is a measure of precision, which indicates how close your mean is to the actual mean. In other words, if you repeated this experiment dozens and dozens of times and calculated the mean of that, how close is your mean to that actual mean? And this is something that can be calculated mathematically. Here's the equation. And so you basically just plug in the standard deviation and you divide it by the square root of the number of trials. And so again, you could try and calculate this on your own. And if you do, you should get these answers. And so what you do next is you then take the standard error and you have to multiply it by 2. So trial 1, the standard error is about 2 times the standard error is about 1, whereas here 2 times is about 4. And so you would add the error bars. You'd go up 1 and down 1 here. And then you'd go up 4 and down 4 there, which means that trial 2 does have a lot more variation. It's so annoying. Than trial 1, which is important information to say. So these would not be considered the same set of data because the standard deviation and standard error are different. So, and then of course, since they do have the same mean and their error bars overlap, these would not be considered statistically different from each other. So that's pretty much it. So here's another video if that kind of goes through this, if that would be helpful for you. And then just a rehash on all those science practices that we have been going through. So that's it. Thank you.